Hi everyone, I welcome you to my series of lectures that is Understanding Pharmaceutical Science with Dr. Hariharan. So today's topic we are going to discuss about immunity and the types of immunity. So in this what we are going to learn is about first what is meant by an immunity and what are the different types of immunity. So first we understand what is an immunity. So immunity is nothing but a state of protection of our body against the foreign organisms, that is the infectious diseases and cancer. So our body is to defend against the various foreign substances that is entering into our body, like viruses, bacteria or any foreign chemicals which may be a protein in nature. So against this our body will defend it not only with these infectious diseases, our body also defends against any tumors, that is the cancer cells is produced in our body. So we can collectively call this mechanism as an immunity. So our body's defense mechanism, especially immunity, can be either a less specific in nature or sometimes more specific in nature. Based on this only we can classify the immunity. So if you see the various types of immunity, immunity can be broadly classified onto two types. One is based upon the mechanism, how it is protecting our body. Second, we can also classify based on how we acquire an immunity. So first we understand the type of immunity based upon the mechanism how it is happening in the body. So we can classify this immunity broadly into two types. One is the innate immunity, second is the adaptive immunity. So innate immunity is the first line of defense against the foreign substances, which is generally non-specific in nature. Whatever foreign substances will enter our body, this defense mechanism will provide our protection against it. If this defense system is overcome by the foreign substance, then the second type of immunity comes into play, that is adaptive immunity. So adaptive immunity is the second line of defense against the foreign substance. This immunity will be very specific in nature. It is highly specific in nature. Even it can recognize an individual foreign antigens, and also it keeps in memory, so that once it encounters for the second time, it gives the protection against that foreign substance immediately. So we can broadly classify the immunity into two types, that is an innate immunity and second is the adaptive immunity. So if you see an innate immunity, the innate immunity is generally composed of components which are present throughout the life of a person, and these components will provide a disease resistance mechanism against the different varieties of foreign substances. And some of the components may be work as a barrier, for example like the skin or the mucosal membrane, like that, and anatomical or physiological barriers, whereas some are in cellular or molecular components, and these sometimes not need to be non-specific in nature. It will be very little bit less specific in nature once it encounters the organs pathogens very frequently. And these components generally includes the macrophages, neutrophils, natural killer cells such as, and macrophages also. These are present along with the barrier of the skin and the mucosal membrane and produce certain antimicrobial components such as mucus secretion. Sometimes our physiology like gastric acid secretion also considered to be a part of the innate immunity. So if we consider an adaptive immunity I already told is the second line of defense mechanism, and it is highly specific in nature. So this adaptive immunity will recognize and selectively eliminates the individual foreign substances, whether it may be a microorganisms or a chemical molecules. Even a small change in its sequence also will be identified by the adaptive immunity. So how the adaptive immunity works, if we see, it contains certain key contributes. That is, one, it is highly antigenic specificity. So to understand the antigenic specificity, first we understand what is meant by an antigen. So antigen is nothing but a foreign substance which may be a living or non-living thing in nature, and it is generally chemically made up of either a protein or an oligosaccharide. So whereas the lipids and polysaccharides does not contribute to be an antigenic in nature. So antigens are 99 98.9% it is protein in nature. So protein is nothing but a sequence of amino acids. So since the adaptive immunity is highly antigenic specific, it can recognize the sequence of amino acid which is present in an antigen. So this recognizing part by the cells present in our body we can call as epitope. So epitope is nothing but an antigenic determinant sites. So what our cells, adaptive immunity cells, especially the lymphocytes, B and T lymphocyte, will specifically identify each individual antigen even though it is different in single amino acid. Second, our B lymphocytes will produce different types of antibodies. So if you see the antibodies, especially the immunoglobulins, carry a variable region which is specifically binds with the antigen. So even a single amino acid difference our body can recognize it. So similarly millions of antibodies can be produced by our body. So we call that our antibodies produced our body are diversified in nature. Each antibodies will be specifically can counter a particular pathogen or an antigen. And third is that it can keep in immunologic memory, that it can produce memory cells. For example, we are doing vaccination to the children. So once we vaccinate a child, what happens to the child is he never gets an infection in a lifetime. That means during the vaccination in the early stage, the immune system will produce an memory cells. These memory cells will be relevant throughout the life of the children, so it protects against the infection for the second time. And final one is that since the antigens are protein in nature, our body cells also contains proteins on its surface. So there will be a need for these cells to recognize which is our own protein and which is the foreign protein. So our lymphocyte cells will recognize both self and non-self. So these attributes makes the adaptive immunity will be very perfect in nature, and it protects against almost all the types of foreign substances which is entering our body, whether it may be a virus, bacteria or any chemical components. So if you can see from the slide, I can divide up this immunity and cell and molecular components into two types. That is the cells associated with the innate immunity. So innate immunity generally protects against the foreign substance with epithelium barrier. Otherwise, the underlying epithelial barrier it carries certain phagocytic cells which may be a macrophage or dendritic cell or natural killer cell, and also it can stimulate a complement system. So once you see an adaptive immunity, the cells which is generally associated with it is the lymphocytes. So the lymphocytes can be a B lymphocyte which stimulates the humoral mediated immunity, and second is the T lymphocyte which stimulates the cell mediated immunity. So the B lymphocytes generally produce an effector molecules, that is antibodies, we also call as immunoglobulins. Similarly, the T lymphocytes will produce a cytolytic T lymphocytes or the helper T cells. In this way, the cells and molecules generally will help in innate and adaptive immunity. So if you see an adaptive immunity which can be further classified into two types. So one is the humoral mediated immunity and second is the cell mediated immunity. So what is meant by an humoral mediated immunity? If you see that if the foreign substance, the antigen, is extracellular in nature, that means outside the cell. So outside the cell means the only part which is present outside the cell is the blood. So blood in Latin term we call as humor. If the foreign substance is present in the humor, that is the blood, what happens is that our B lymphocyte will produce an antibodies. These antibodies will effectively eliminate this foreign substance in the blood. So it is called as humoral mediated immunity. So next is if you see the cell mediated immunity, in these conditions the foreign substance has entered into a cell. For example, consider a virus. Virus might have entered inside a cell. So what happened is this infected cell should be completely removed. This can be done by the T lymphocytes. So T lymphocyte can itself converted to a cytolytic T lymphocytes, or it can stimulate a macrophage. Both the things what it will do is it is engulf the infected cell. So that is called as phagocytosis. So it will destroy the microbes infected cells by the T lymphocyte, is generally called as a cell mediated immunity, because it removes the foreign substance which has infected the cell. So adaptive immunity can be broadly classified into cell mediated immunity and humoral mediated immunity. So majorly the classification, we can further classify the types of immunity into second division. If you see the types of immunity, one is based upon the mechanism how our body is defending, that is the innate and adaptive immunity. The adaptive immunity is further divided into humoral and cell mediated immunity. And second type of classification, we can classify based upon the acquired, how our body acquires the immunity. So if you see the acquired immunity, that is how our body develops these antibodies, or whether we need the antibodies from outside. So we can classify this into two major types, that is called as active immunity, and second is the passive immunity. So both this active and passive immunity can be further classified based upon whether it is occurring naturally or it should be artificially induced. So if you see what is meant by an active immunity, so active immunity is nothing but an active involvement of our body. That means our body once encounters a foreign substance, it can synthesize the antibodies. Then it can also synthesize the immunocompetent cells like B lymphocyte, T lymphocyte, or natural killer cell. So it can eliminate the foreign substance by itself. It is called active participation of our body, or actively producing it, defending against the foreign substance. Whereas once I consider the passive immunity is nothing but the administration of the antibodies or the immunocompetent cells from one host to another. That means delivering of the monoclonal antibodies from outside to a patient is a type of a passive immunity. So this is the basic idea what is meant by an active and passive immunity. If I classify the acquired immunity broadly into two types, that is active immunity and passive immunity, then I can further classify it based on whether it is naturally occurring or artificially produced. So the active immunity can be classified into naturally active immunity and artificially active immunity. Similarly, the passive immunity can be classified into naturally passive immunity and artificial passive immunity. If you see the natural active immunity, it is generally produced by the person during its course of life. Once he plays in a mud, he may be exposed to a small microbial load. So what happened is the body started defending against the infection by producing the antibodies. It is called naturally producing immunity, naturally active immunity. If you see an artificial active immunity, we are inducing the body to produce antibodies by means of vaccination. So by means of immunization, we administered a small quantity of attenuated bacteria or virus, or killed bacteria or virus. Thereby what happens is our body gets stimulated. Once it encounters the foreign substance, our body has a caliber to defend it. This is called as artificially active immunity, because our body will produce the antibodies against that foreign substance. If you see the passive immunity, the natural passive immunity, the best example is that once the woman is pregnant, the fetus will get antibodies from her mother. That is, the transfer of antibodies from mother to the fetus through the placenta is the naturally passive immunity. If you see the artificial passive immunity, nowadays what we are doing is, if a person gets infected with a disease and he is no more able to produce the antibodies, we will produce the antibodies needed for the treatment in large quantities. Then we administered it. So this can be done by producing the antibodies, monoclonal or polyclonal antibodies largely, or we can also do infusion from the infected patient. It's plasma therapy, we can call it, we can do it. So this is an artificial immunity. So this is the basic idea about the immunity and the different types of immunity. So I thank you very much for your support. So in future I will discuss each immunity in an elaborated manner. Thank you very much.